Wonderful. Start to see attendees arriving. Um, I really thank everybody for taking some time out of their summer to come and enjoy the Lipid Research Division virtual seminar series. Today we've got two great talks that are focusing somewhat broadly on the concept or idea of mitochondrial membrane remodeling, going from some exciting new studies that can maybe make its way into the clinic, as well as some really detailed mechanistic insights about how this super mitochondrial lipid, cardiolipin, can help to remodel the inner mitochondrial membranes. So, without further ado, we've now got ourselves up to a steady attendance count. So, I'd like to first hand off to Dr. Sanna Masud, and she's going to tell us a really interesting story about, again, how we might have some clinical impact with treating some devastating human syndromes that are affected by mitochondrial membrane lipids.
Great, thank you. Let me just share my slides. Alrighty. So thank you again for the introduction, Josh. So I'm a postdoctoral research fellow at the Hospital for Sick Children located in Toronto, and I'm in the lab of Dr. Jason Moffat. And today I'm going to give you a bit of a highlight reel on how we identified a rare genetic suppression interaction by genetic interaction screening and how it impacts a rare mitochondrial disease. So I put my email here. I've also put it into the chat. So please feel free to email me if you have any questions that we can't get to today or potential collaborations and so on. So for our lipids research community, I don't think cardiolipin needs much of an introduction. It is one of the most unusual and functionally important phospholipids in the cell, very unusual in the context of it being a dimeric structure that no other phospholipids seem to have, and it being exclusive to the mitochondria and predominantly within the mitochondrial inner membrane. So that's how it's gotten the title of the signature phospholipid of the mitochondria. So here it helps organize the respiratory chain, shape cristae architecture. It'll regulate mitochondrial dynamics as well as coordinate stress responses. So what I've shown here is also a linear representation of the pathway. So it is a bit more complex than this, but the part that we're really wanting to focus on, if I got my pointer here, is kind of the terminal component of the pathway where we're looking at nascent species and the intermediate monolysocardiolipin and how it gets converted to mature cardiolipin, with a strong focus on the enzyme TAFAZZIN, or known as TAZ, that is responsible for generating mature cardiolipin so that it can exert all of these functions. But despite all this work that we've done on cardiolipin biology to date, we still have an incomplete understanding of how cardiolipin metabolism is buffered, remodeled, or rewired in the context of human disease. And this question is especially urgent in the context of Barth syndrome, which is a rare mitochondrial disease caused by mutations in the gene TAFAZZIN. And it's an X-linked recessive disorder, so only affects males, but it's got these pleiotropic clinical manifestations that also make it quite difficult to diagnose. So we see various temporal onset of phenotypes in these patients, but we also see a full spectrum of types of phenotypes, making it quite difficult to understand the etiology of the disease. And here, the loss of TAFAZZIN essentially is going to disrupt cardiolipin homeostasis. So it creates a buildup of the intermediate monolysocardiolipin and causes a decrease in mature cardiolipin species. And this results in profound mitochondrial dysfunction phenotypes. And this is also one of the ways that the disease is diagnosed. Aside from sequencing to find out if these patients have a TAZ mutation, you could do blood spot assays to see that the ratio of monolysocardiolipin to mature cardiolipin becomes disproportional. And we'll talk a bit more about this going forward. But as I mentioned, the clinical manifestations of Barth syndrome are quite pleiotropic, but this is true for many mitochondrial disorders. And I'm kind of pointing out here even monogenic Mendelian disorders like Barth syndrome, where you know what the perturbation is. So here, a mutation in TAZ, but how it's clinically presenting is quite diverse with central nervous system phenotypes, immune phenotypes, skeletal phenotypes, and cardiac phenotypes being more dominant since they're tissues with higher energetic demand. But this really mechanistically raises a broader question on when a core lipid remodeling pathway, such as the cardiolipin pathway, fails, what other genes and pathways can determine whether a cell will adapt to this new feature or collapse. So this is where our lab looks at genetic interactions and how genetic interactions are a powerful lens to look at these problems. So rather than studying genes one at a time, genetic interaction mapping asks how the effect of perturbing one gene changes in the context of another. And it lets us identify interactions and pathways that could buffer, worsen, or suppress these disease-relevant defects. So you got two types of interactions at play. You have negative genetic interactions where the two mutations can essentially cause cells to either be synthetic sick, so you'd have a proliferative defect, or synthetic lethal, where they're no longer viable and drop out of the population. Or on the other spectrum, you could have positive interactions where you'd have some type of growth advantage. And you can have these masking interactions where it's not kind of a complete suppression, but suppression interactions, which is what everyone would essentially want to look for in these disease contexts, but they're very rare to find. So for lipid biology, this is a particularly exciting way to look at this problem because lipid metabolism is highly networked. You've got enzymes, transporters, remodeling reactions, organelle contacts, and stress responses that all act together to determine the final lipid landscape of the cell. So the framework that we use is essentially we're asking a simple but challenging question of can the genetic dependencies of TAFAZZIN deficient cells reveal new regulators of cardiolipin metabolism, any alternative pathways that might be at play, and eventually the opportunity to find a therapeutic. And the way we do this is by genome-wide CRISPR screens. So very briefly, we use the Toronto Knockout version 3 guide RNA library developed in Jason's lab. That we've got 4 guides targeting every gene in the human genome, or all protein coding genes. And then we use hap1 cells as our kind of model cell line. It's a near haploid cell line. And the way we set up our screen is the TKOV3 library is a lenti library. We'll transduce cells at a multiplicity of infection of 0.3 so that we aim to have 1 viral integrant per cell. And when you're looking at hap1 parental cells, this will tell you single mutant fitness effects per gene perturbation. But in a query cell line, so in a TAZ knockout cell line, as an example, now we can look at all the combinations of gene A, with TAZ, all combinations of gene B across the human genome and what the double mutant effects are. So our screens, we take a T0 reference population where all guides should be represented in that population and collect genomic DNA. And then we take our screens for about 15 cell doublings and then again harvest genomic DNA and then do next generation sequencing to compare the populations or the guide RNA enrichment or dropout. And then we've got computational pipelines where we'll do all of our analyses and QC. And then we've generated this quantitative genetic interaction score that really helps us parse out positive interactions compared to negative interactions in our query cell lines compared to what we know from single mutant fitness in our hap1 parentals. So we use this pipeline to figure out what are the genetic modifiers of TAZ. And with an industry collaborator, Scenic Biotech, we use 2 orthogonal approaches for our screening platform. They did gene trap mutagenesis screens, and then we did the CRISPR GI screening pipeline that I just described. So I'm going to focus a bit more on this side of the plot, but in this scatter plot, what you're looking at are the genetic interactors of mutant TAZ. And in yellow are our positive genetic interactions, and in blue are our negative genetic interactions. And then the colors depict, these are 5 biological replicate screens done in different clones, media conditions, and so on. And we get this robust profile, and the darker of the 2 colors is essentially telling you that it's come out in how many screens. So here is where the agreement between our 2 screening platforms and where we started to really look at our top interacting genes. And again, finding positive interactions on what could mitigate the effects that we see of Barth syndrome is what we were really interested in. And our top interaction was this gene ABHD18, but in 2017, it was called C4ORF29, so it had no annotation whatsoever. So we began to explore what does this ORF do. So first I generated these clonal knockout cell lines. and just ran a proliferation assay, and this would validate our screen findings. And we could see that when we knock out ABHD18 in our TAZ deficient cells, we've restored the proliferative effects back to wild type levels. I had also done a CRISPR screen in the ABHD18 or C4orf29 knockout cells. And what was striking here is we got many of the same genetic interactors coming out as reciprocal interactions, really pointing to that ABHD18 and TAZ may work together or within the same type of pathway. And then looking at publicly available data with DepMap and based off of mRNA expression, when we look at deviations in TAZ expression, the top modifier tends to be ABHD18. And again, we get the reciprocal where with ABHD18 perturbation, we see the top modifier coming out as TAZ. Now, again, we knew nothing about this ore. So is it even a mitochondrial protein? So that was one of the first experiments that we had done by tagging ABHD18 and then just doing a live cell imaging experiment with mitotrackers. We found good co-localization agreement. And then we'd also done bioID on this by tagging both the N and C terminal ends and then growing the cells in either glucose, which I'll call kind of a permissive condition here, or galactose to enrich for mitochondrial interactions. And then we saw that a significant proportion of the interactions were proteins localized to the inner mitochondrial membrane, specifically components of the respiratory complexes, but also found other transporters and mitochondrial proteins involved, leading us to hypothesize that ABHD18 is a mitochondrial protein that might reside in the inner mitochondrial membrane, similar to TAFAZZIN. Now, this is where it gets really exciting because I told you that the key kind of metric to look at Barth syndrome is that MLCL to CL ratio. So when we performed lipidomics on our cell lines, we could see that we were starting to restore that imbalance in monolysocardiolipin to cardiolipin in our double knockouts compared to see the enormous buildup of MLCL that we get in TAZ knockout cells. But we also started to see that in the ABHD18 knockout cells, we start to develop more nascent cardiolipin species and really low levels of monolysocardiolipin species. So then we got thinking that, okay, maybe ABHD18 is actually contributing to the production of monolysocardiolipin from nascent. And I should also mention that from the name change of C4orf29 to ABHD18, that came out based off of looking at sequencing homology, and that C4orf29 contained this AB hydrolase domain. So that's the ABHD18 component. But to directly assess this, we purified human wild-type ABHD18 protein, and we looked at what we know as the catalytic component, being the serine 199 motif, and then performed thin layer chromatography with different cardiolipin species as substrates and found that tested cardiolipin species, regardless of acyl chain length or degree of saturation, were effectively converted to monolysocardiolipin only by wild-type ABHD18 protein and not the S199 catalytic dead ABHD18 protein. So it suggests that ABHD18 protein is a cardiolipin deacylase. And then I'm also showing a molecular dynamic simulation showing how monolysocardiolipins, and we've tested this with nascent species as well, fits into the pocket of ABHD18 right at that serine motif. So kind of again, reiterating that this is the catalytic component. And this was really important because now we've found what is considered the missing lipase in human cells that converts nascent cardiolipin species into monolysocardiolipin. So in yeast, this is CLD1, but it's remained elusive in human cells to this point. And we've now directly implicated it in how it's functioning with TAZ by essentially feeding it the substrate it needs to further remodel cardiolipin. So we started to test this in mice models, and we did this again with Scenic and Doug Strathy's lab in Scotland. And we generated CRISPR knockout mice for both ABHD18, TAZ, and the double mutant. And all the phenotypes I'm going to show you are based off of male mice cohorts, since they're the ones that developed the Barth syndrome phenotypes. And immediately we could already see what was quite striking was we were rescuing the Mendelian birth ratios for male mice with our double knockout mice. We also were able to restore the weight of TAZ deficient mice, or now with our double, which is also characteristic of what we see in Barth syndrome patients. Barth syndrome patients are also sterile. And with our mice, we were able to restore fertility. So we're able to actually generate sperm that we were able to then further mate our mice and generate cohorts. So this was quite nice, and we saw no gross abnormalities, and really indicating that we've got complete phenotypic suppression at play. Cardiac stress markers we were looking at, so things that are typically profiled in Barth syndrome patients. So by doing qPCRs and looking at fractional shortening in the mice, again with our double knockouts, we were able to restore pretty much back to wild type levels. We also performed lipidomics on these mice hearts, and similar to what we saw in our HAP1 cells, we're starting to restore that monolysocardiolipin to cardiolipin ratio buildup. And again, the same types of cardiolipin species that we saw in HAP1 cells, we're observing in the mice hearts as well, that we're getting a shift to these more nascent saturated species. We did transcriptomics on these mice hearts, and again, also saw gross rewiring that occurred in TAZ knockout mice that was essentially restored in our double knockout mice. And you can see that here in the plot here that we almost rescued all of the transcriptional effects that were associated with the TAZ knockout. And then further, we wanted to see, can pharmacological inhibition match what we're doing genetically? So we started working with another industry collaborator, Lundbeck, and developed a small molecule covalent inhibitor that is known as ABD646. So the first thing we also wanted to assess was, okay, how do the mitochondria look when we're pharmacologically inhibiting ABHD18? And we did these experiments too that you could see in the paper for the genetic models. But upon putting ABD646 on HAP1 cells, we started to see a restoration in cristae structure that was not present in our HAP1 TAZ mutant cells. We also characterized this by fitness to see that as we were dosing ABHD18 on our cells, we were starting to restore the proliferative effect that we would see on our HAP1 cells on their own. We then took Barth syndrome patient fibroblast cells and did the same experiment. So here's, you can see with the TEM images, it's quite striking what we see with these really swollen looking mitochondria. We now get these more dense, I'm assuming cristae packed mitochondria upon ABD646 treatment. And this is only within 96 hours of treatment. And we already start to see again quite a proliferative bump with these cells. So we also wanted to look at heart phenotypes, and we used a zebrafish model to do this. So in collaboration with another lab here at Sick Kids, Ian Scott's lab, we started generating TAZ mutant embryos using morpholinos. And here you can see what a wild type zebrafish heart looks like. And these are a transgenic line that are EGFP tagged for MIL7. What ABD646 treatment looks like on the heart, and then what the TAZ morpholino treatment looks like. So you get this giant edema and like this linear heart phenotype, but that we can restore that upon ABD646 treatment within 3 days. So this is like quite a striking result. I do have a video here. They might play a bit slowly, but not only were we rescuing the morphology of these hearts, we were also restoring their beating potential, which was quite amazing for us to see. And I think the last component that I really want to highlight in this talk is something that I'd always wondered was, cardiolipin is so essential. If TAZ is the kind of last step within this pathway that remodels cardiolipin for all of its functions, how do we even get Barth patients to begin with? And if you were to look at something like DepMap data, it is considered a strongly selective essential gene. So how does that work exactly? And what we found was TAZ activity or its expression is directly correlated with ABHD18. So we took our HAP1 mutant cell lines, and with Hilary Vernon's lab, we got iPSC and cardiomyocytes or RNA sequencing data from them. And then we also looked at our mice data. And as soon as you had TAZ perturbation, you had this concomitant ABHD18 perturbation. So essentially something in the cell is causing or sensing this monolyso buildup and trying to drive ABHD18 activity down, probably to mitigate the flux of the pathway. It just doesn't get down regulated enough where there's no phenotype. So this again is where ideally our covalent inhibitor becomes an actual therapeutic for these patients. And the last slide that I've got here is we've benchmarked this with the newly approved or FDA approved treatment for Barth syndrome being elamipretide. So elamipretide binds to cardiolipin and promotes aggregation of cardiolipin molecules, but it's not really hitting the exact point or is not as targeted as inhibiting something like ABHD18, which is responsible for that buildup to begin with. And at significantly lower ABHD18 or ABD646 concentrations, we're again able to restore the proliferative effects compared to escalating doses of elamipretide in a significantly shorter amount of time as well. So we are really excited about this and are working quite hard to develop this further as the first targeted therapeutic treatment for Barth syndrome and really showcasing how genetic interaction mapping can identify suppressors for this rare disease. So there was a lot of people who helped me along the way for this project, kind of illustrating it here and our funding sources. And for anyone who's not familiar with TAZ and hasn't encountered why it's called TAFAZZIN, I have put TAFAZZI in here, which is what the gene was actually named of. And one of our students is an artist who's now made a rendering for me, showing the collaboration between ABHD teen, who I'm calling Antonio, with TAFAZZI. So with that, I'd be happy to take any questions.
All right, fantastic. Really exciting talk. So we'll go ahead and look at the attendees for any hands raised for questions. So I have first Subrajit Banerjee. I'm going to go ahead and allow you to talk, and you can go ahead and unmute yourself.
Okay. All right. Thanks, Josh. It's a beautiful talk. Really, really enjoyed it. Um, so I have basically two questions. One of them is like, so this means that all our cells, like especially the terminally differentiated cells, like our body should have very, very low levels of ABHD18. Otherwise, you'll convert all cardiolipin to monolysis cardiolipin, right? And the second question that I have is like I saw another exciting hit, which is STAR-D7. Like, do you have any insights on like because that's a start domain containing lipid transfer protein,
Yeah.
right? So do you have any insights on how that is working together? Uh,
Yeah, so in regards to ABHD teen expression, it's very similar to what we see for TAZ expression across all tissue types. We are working on figuring out like the half-life of the protein and how long it's
uh,
actually sticking around for, but it's really like in normal cells, you never see, it's hard to detect even by lipidomics an actual amount of monolysocardiolipin, so the
gotcha.
pathway goes quite efficiently. Um, so I don't think that there's any concern about elevated ABHD teen expression levels as long as you've got functional TAFAZZIN that's constantly converting the product that ABHD teen gives you. This is why we only
Uh,
really see the issue when it comes to Barth patient, like TAZ mutant individuals. For STAR-D7, yes. So one, I can tell you it is not a suppressor the way that ABHD teen is. So it is the sole phosphatidylcholine transporter to the mitochondria, and it's actually by fitness. So if you were to look at it on DepMap, is considered more essential than TAZ. So we have been doing quite a bit of work on the relationship between STAR-D7 and how it's implicated in the cardiolipin pathway. And hopefully our paper on that will come out soon. So I can't totally say very much more on
okay.
that now, but it's not suppression the way ABHD teen is.
Very cool. Look forward to it. Thank you.
Now, before we get to the next question, I don't want to twist your arm, but Thomas Longer did have a really nice paper about STAR-D7 regulating coenzyme Q
Mhm.
biosynthesis. Is that maybe an angle you're chasing?
No.
Okay, all right, good to know. All right, so now on to our next questioner, which is my favorite questioner, Yoon Chu Kim. You can go ahead and unmute yourself.
Hello?
Oh,
You hear me?
yeah, we got you now, Yunju.
Yeah, okay. Thank you. So it was really fantastic talk. Thank you. I'm wondering, I know the LCLAT, that's the another acetyltransferase protein, like a TAZ. So for ratio mature and immature cardiolipin. Did you get any pickup from your screening related to even this ABHD, your protein?
Yeah, no, that's a great question. That and the MLCLAT1, we don't see it come out significantly in either screen format. So I know, I guess before we identified ABHD teen, what was really generating monolyso that would then go to become mature cardiolipin or feed TAZ was kind of elusive. People were really wondering, is it PNPLA8, IPLA2? So we don't see those coming out as significantly whatsoever in our screens. I don't negate that they are probably contributing to some conversion. It just clearly does not come close to the effects that ABHD teen or TAZ have.
Okay, do you know whether this LCLAT or TAZ is more dominant for cardiolipin level or not? Or do you know about
Yeah, so I think ABHD teen and TAZ are the two most dominant members of the cardiolipin pathway.
Okay, all right. Thank you so much.
Great. So now we'll move on to Fred Posmeyer. I'll go ahead and you can unmute yourself.
Okay, well, it's clear that if you have too much monolyso cardiolipin, that's bad. But you do have some. And normally, is there why would that get formed? Is there a function for the monolyso?
So you mean even in the presence of an ABHD teen knockout?
Sure, whatever.
Yeah, so I mean, I feel like Dr. Khalil will hopefully be able to answer that a bit better for me for the biochemistry of really why monolyso is the intermediate before generating mature cardiolipin. Yes, we can generate trace amounts of monolyso even with our perturbations that we've seen. Again, maybe that's partly in play from IPLA2 or PNPLA8 that is trying to remodel the cardiolipins in the absence of either one or both of our enzymes. But essentially monolyso, why is it an intermediate? I'm not entirely sure how to answer that. Like, why is cardiolipin dimeric too? How is that? I mean, I know it's involved for generating the curvature of cristae, but I guess in order to alter the saturation levels before it can effectively do that, monolyso has to be generated.
Okay, so it's really a case of changing saturated to unsaturated fatty acid or vice versa.
Yeah.
Okay, that makes so. That is a function, I would say.
Yeah.
Thank you.
Yeah, it's interesting to postulate on the role, whether it's a sink for fatty acids, what actually is happening during these remodeling steps. I know there's some fantastic work in yeast that has pushed a lot of our thinking on cardiolipin. So I think with that, we're coming up on the halfway mark of the hour. So I'd like to thank you again, Sanna, for a fantastic talk.
Thank you.
Oh, and I don't know, Fred, if you wouldn't mind to mute yourself.
I'm trying to see how to do that. Can you do it?
I'll see if I can get you. Yeah, I think I can. There we go. Okay, the classical music is now gone, as much as it was enjoyable. So I think transitioning to the next talk, as was alluded to by Sanna, I think what has happened here in the laboratory of Dr. Halil Aden is they've really pushed the boundaries of being able to understand what cardiolipin does, and especially the different molecular species of cardiolipin. So without further ado, I'll hand it over to Halil to tell you about his fantastic structural work.
Thank you, Josh. And it was a great talk, Sanna. Thank you. Yeah, my name is Halil Aydin. I'm an assistant professor at NYU. And we have a long-standing interest in understanding mitochondrial function because it's becoming increasingly evident that this organelle actually is not just a powerhouse of the cell, but it functions as a cellular and organismal signaling hub. Mitochondria uses specific signals such as metabolites, lipids, and proteins to communicate with its environment. And these communication pathways result in regulation of metabolic events, stress response, extracellular communication, and cellular differentiation. And my lab is interested in understanding conserved pathways that control cellular communication and decision-making processes. And we want to dissect the role of these molecules in mitochondria to understand how they control this organelle. And as you all know, mitochondria is a multifaceted organelle. It has many proteins and protein complexes that perform essential functions for cells. Many of these processes are localized to inner membrane, and their function is directly related to a membrane composition, particularly the presence of cardiolipin. But what's, I think, interesting about mitochondria is that currently we know that there are 1,200 or so proteins that are localized to mitochondria, but around 20% of them has no known function. So these proteins localize to this organelle, but we don't know what they specifically do. And the estimated proteome of mitochondria is around 1,500 proteins. So we think that there's still a lot to uncover about mitochondria and how it regulates essential cellular pathways. And another interesting thing about mitochondria is that its function is ultimately linked to dynamic changes in its morphology. And these morphological changes can influence human health and disease. In fibroblasts, for instance, mitochondria form these long dynamic networks, and these mitochondria frequently fuse and divide and adopt divergent of morphologies. They also form clusters, they form nanotunnels to communicate with each other, they move long distances, and they interact with other organelles. So these dynamic remodeling of this organelle network is referred as mitochondrial dynamics. And over the years, a few proteins implicated in controlling mitochondrial form and function. Here, I'm showing you the proteins that are related to this talk. There are actually other machineries that perform some of these functions. But for dynamic fusion and fission events, we have a few dynamic-related proteins that controls the morphology of this organelle. On the outer surface, we have DRP1, or dynamic-related protein 1. It's a cytosolic protein, but it recognizes these receptors, FIS1, MFF, MIT49, and MIT51 on the mitochondrial surface, forms these ring-like assemblies on the surface, and catalyzes the fission of the organelle. And for the fusion, we actually have 3 different proteins that catalyze 2 separate membrane fusion events. Mitofusin 1 and mitofusin 2, these are membrane-anchored proteins that catalyze the fusion of outer membrane. And inside the organelle, we have OPA1, or optic atrophy 1, that facilitates the fusion of inner membrane, but also contributes to the regulation of CRISPR architecture. So a few years ago, we were interested in understanding how human OPA1 may catalyze the fusion of mitochondrial inner membrane. And we knew that to answer this question at the molecular level, we really need a reductionist approach where we go to the basics and we have a full control of our system. So to do this, we developed a reconstitution assay. And in this reconstitution assay, we used human OPA1 and we reconstituted with synthetic lipids or liposomes to dissect the molecular basis of protein-protein and protein-lipid interactions. And this is a very simple system. We can make liposomes with different lipid compositions. And when we incubate these liposomes with our OPA1 protein, we observed that human OPA1 can form these ordered assemblies, or it can facilitate the tubulation of these membranes. So this was a very simple but very at the same time useful system, because this showed us that our construct is functional. It can remodel membranes, but it also allowed us, it provided a good platform for us. So using this system, we actually image these reconstituted samples with cryo-electron microscopy, and we determine the near atomic resolution structure of human OPA1 assembled on membranes. And just like we predicted, OPA1 forms these well-ordered assemblies on membranes, and the molecules form this helical assembly that wraps around the membranes. And just like the other fusion and fission proteins, OPA1 belongs to the dynamic superfamily of proteins, and our structures show that it has a topology similar to other dynamic related proteins. And using these maps, we actually build a molecular model for human OPA1. And as you can see here, it has an N-terminal GTPase domain, followed by a bundle signaling element, BAC domain, a stalk region that's important for oligomerization, and a lipid binding domain, which we call the pedal domain. So this was very exciting for us, but to me, the most exciting part about this research was we can now actually study how OPA1 interacts with membranes and modulates the architecture of mitochondria. And our structure actually unexpectedly, I would say, revealed that the pedal domain of OPA1 form tight interactions with the membrane, and part of the pedal domain is actually inserted in the membrane. So this is very unique. We predict that, for instance, viral fusion proteins function this way. They have a fusion peptide that's inserted in the membrane, but this was never been observed with a near full-length protein. So these are actually the first structural examples of how a fusion peptide might engage with the membranes. And if you look at our maps closely, we see that there are actually two regions that engage with the membrane. One we call the docking region, which contains these arginine 857 and 858 residues. And then the other part is the membrane inserting loop region of the protein, where a loop is actually embedded in the membrane. We actually determined the structure of this assembly in two different conformational stages. In the first conformation that I just showed you, the protein layer actually sits on top of the membrane, and the pedal domain is embedded in the membrane. So the protein layer forms strong interactions with the membrane. And this assembly had an inner diameter of around 19 nanometers. In the second conformation, what we noticed is that this inner diameter was constricted, so the membrane's shape changed, but also the protein layer moved away from the lipids, and it was forming only minimal contacts with the membrane. In the second assembly, we saw that membrane inserted loop was pulled away or pulled out and away from the membranes, and there was just minimal contacts mediating interactions or shaping membranes. So based on these structural analysis, obviously we have done many other experiments, but we came up with this model showing that how human OPA1 may facilitate membrane fusion. In our model, we predict that the long isoform of OPA1, which is membrane anchored, initiates membrane remodeling, and these long isoforms then basically recruit short and soluble form of OPA1, and together they form these scaffolds that engage mitochondrial inner membrane. Then the energy provided by the oligomerization of these short and long isoform creates this curvature stress and further tubulates these membranes, and eventually these unstable tips of these membranes fuse, and then the whole mitochondrial inner membrane fuses. So this was great. It really provided us a mechanism about mitochondrial inner membrane fusion, which is turns out that it's very different than other fusion mechanisms. But there was one thing missing here, and that missing part was, what is the role of these lipids in this process? Because we knew that OPA1 has to engage with the membranes to remodel them. So to answer this question, we went back to our reconstitution assay, and we prepared liposomes with different lipid compositions. And in these experiments, we screened a few different lipids and realized that in the absence of cardiolipin, OPA1 cannot bind and remodel these membranes. And we can quantify these assays. And here I'm showing you the quantification of this. And you can clearly see that only in the presence of cardiolipin, OPA1 can remodel membrane. So it shows that there's a very strong specificity towards cardiolipin. Although we observed some remodeling with POPS, that activity was 95% or so less than cardiolipin-containing liposome. So we are very confident that it's not the negative charge of cardiolipin, but it's actually cardiolipin itself is critical for this process. Obviously, this also opened up other questions, research questions for us. And one of the questions is what type of cardiolipin? Cardiolipin has many different species, but which ones are important for regulating these dynamic membrane remodeling events? And to answer this, we prepared liposomes with 2 saturated and 3 unsaturated cardiolipin species and tested their effect on OPA1's activity. First, we looked at membrane binding, and to our surprise, what we saw that OPA1 was binding to unsaturated cardiolipin species effectively, but this activity was decreased with saturated cardiolipin species. So at the binding stage, there was a preference. We also, again, looked at the remodeling, and we saw a similar pattern in the presence of unsaturated cardiolipin species. OPA1 was able to remodel and elongate these tubular structures, but in the presence of saturated cardiolipin species, OPA1's activity was decreased. So this was great. Now we know that not only the cardiolipin, but it's actually the unsaturated cardiolipin species are critical for the function of these proteins. We knew that OPA1 is selectively engaged with cardiolipin, but we still don't know how does this happen. And unfortunately, many of our experimental tools do not allow us to answer this question directly. The reason is many protein-lipid interactions occur at a very short time and length scale, and our experimental methods are not good enough to observe these processes. So we decided to opt in with a computational approach and teamed up with Matteo Del Perero's lab in Switzerland to develop MD simulations to study OPA1 membrane interactions. And in these simulations, we used an OPA1 tetramer as a representative basic building block of this assembly. And we noticed that within actually a few nanoseconds, in these membranes, cardiolipin really moves near protein contact sites and accumulates near OPA1 membrane contact sites, forming direct interactions with these proteins. The major advantage of this tool, in my opinion, is that you can rewind and replay these movies. You can simplify them to analyze them, and you get a lot of quantitative information. And that's what we did. We removed all the other lipids in the membrane, and we just visualized cardiolipin molecules in these membranes. And what we noticed was very interesting. Although cardiolipin molecules resided at this protein contact size 5 times longer than any other lipid, they formed short-lived interactions. These weren't long-lived interactions. So as you can see in this video, the lipids engage with the membrane interacting regions of the proteins and bounce between side chains and then slowly diffuse away, but they still reside there 5 times longer than any other lipid. And that's what our quantifications show, that this is the average cardiolipin density near protein contact size. And like I said, we can also look at the interactions at the residue level. And then these analyses actually showed us that there are really 2 key regions. One is the membrane inserting loop region that forms several interactions with cardiolipin. And the other one is the docking region that I showed you earlier. And these interactions are much longer and stronger compared to POPC and POPE. So using this information, we mutated these residues and tested them again in our reconstitution assays. And our experiments show that when we mutate these residues, we do not see membrane binding and remodeling activity of OPA1, showing that this interaction between cardiolipin and these two regions are very specific and essential for protein's activity. At this point, we had a good amount of molecular insights about the function. So we wanted to push this further, and we wanted to, again, see these interactions experimentally. But like I said, it's very challenging to observe these processes experimentally because we do not have good tools to do that. We took this challenge, and we wanted to understand cardiolipin protein interactions within intact membranes. And in recent years, several groups have attempted to develop different tools to bridge chemistry and biology to study lipid dynamics. And we wanted to use these tools and approaches, but we had two conditions. One was we wanted to have minimal alterations to the chemical structure of cardiolipin. And then the second one we wanted cardiolipin to behave indistinguishably from its unlabeled counterpart. So we decided to use brominated and iodinated lipids to label cardiolipin, and we generated a brominated cardiolipin probe. And this modification allowed us to capitalize its bromine's lipophilicity, steric, and enhance electron scattering properties. And we thought that we can now visualize cardiolipin molecules within intact membranes using cryo-electron microscopy. And we use these brominated lipids and again determine the structure of OPA1 bound to brominated cardiolipin containing membranes. And our structures show that OPA1 engages with the membrane the same way, and this bromination or brominated cardiolipins did not change the protein's activity on membranes. And in fact, if you compare the structures from both models, we had less than 1 RMSD, showing that brominated cardiolipin does not induce any notable changes to OPA1 assemblies on membranes. So using these models, we also analyze the membrane composition. So we can actually, now we have our native or unbrominated membranes and brominated membranes. And when we quantify the density or the scattering from these membranes, we notice that there is an increase in the outer membrane in brominated membranes compared to unbrominated membranes. And we can actually study these two maps and generate a difference map. And our difference map also showed the same thing, that there is some excess scattering in the outer membrane. Unfortunately, cardiolipin is a very dynamic molecule. We weren't able to specifically pinpoint which residues are interacting using this experimental method, but we still show that it is enriched in the outer or OPA1 binding, OPA1 bound leaflet of these membranes, so that we were able to extract some experimental evidence from this. And finally, we wanted to assess whether our structure-based insights can predict protein loss of function phenotypes in living cells. And again, we mutated these residues and we tested their function in living cells. In these experiments, the wild-type protein showed this tubular mitochondrial morphology. And when we introduced those two mutants, the membrane inserting loop mutant or arginine 858 mutant, we observe more fragmented mitochondrial morphology, indicating that these residues that mediate specific interactions with cardiolipin are important for basically modulating or regulating mitochondrial network morphology in living cells. So using this information, then we were interested in understanding how changes in cardiolipin composition can affect human health and disease. And just like Sanna mentioned, we were interested in understanding how MLCL actually causes mitochondrial dysfunction and human diseases. And I'm not going to go into details of Barth syndrome, but we basically wanted to understand the effects of MLCL on protein activity in mitochondria. So to do this, we again started with the computational methods, but to our surprise, we saw almost no difference between MLCL and cardiolipin interactions with OPA1. If you look at this figure, it was almost identical. The residues, the number of contacts were very similar. And when we performed protein binding assays, we also saw a similar pattern where OPA1 was binding to MLCL containing membranes, and we did a concentration screen. We looked at low concentrations of MLCL, 1 to 5%, but we also completely replaced cardiolipin with MLCL at 25%. At that point, we saw a 30% or so decrease, but there was no significant change, and this really puzzled us for a bit. Then we decided to look at changes in membrane deformation or remodeling, and that we saw a difference in our MD simulations. Cardiolipin can remodel, OPA1 can remodel cardiolipin-containing membranes, and here red means that that part of the membrane is pulled, and blue means that it's pushed, so creating a curvature. But these changes were reduced in MLCL containing membranes, and we didn't see as much deformation in MLCL containing membranes. Again, we quantify this in our remodeling assays. And what we saw that even at very low concentrations of MLCL, 1 to 5%, we saw more than 50% decrease in membrane remodeling activity of OPA1. And when we replaced cardiolipin with MLCL, completely replaced it, we saw even higher, around 90% decrease in total remodeled area. Now we can actually tell that how we can actually explain how MLCL is disrupting protein activity in mitochondria, particularly proteins that are responsible for maintaining mitochondrial architecture. It seems like MLCL is changing the malleability of these membranes, and it provides and it compromises proper remodeling of these membranes. So now I think it's time for us to update this model or improve it. And we are thinking about expanding this model. But here's a quick summary. Now we know that actually cardiolipin is enriched in the outer membrane upon protein binding, and this allows OPA1 to remodel these membranes. But in the presence of MLCL, this binding activity is restricted. MLCL is interacting with OPA1 just like cardiolipin, but it changes the malleability of the membranes. It restricts remodeling, and that causes mitochondrial dysfunction. Finally, I just want to show one quick slide. Using this now a foundation on mitochondrial remodeling, we think that it's a good time to study mitocellular communication and investigate how this organelle communicates cellular signals. And there are many molecules actually that communicate with mitochondria, and we want to understand the molecular basis of these processes. And I just want to give you one quick example. We recently teamed up with Dale Abel's lab at UCLA to understand how mitochondria communicate cellular signals. And Dale is a clinician scientist, and he showed that he studied a pregnant woman, and as you know, the estrogen levels increase at the third trimester. But Dale's lab showed that the OPA1 expression levels also increase. And what they also showed that these increased estrogen, especially in platelets, accumulates in mitochondrial membranes. So we then asked this question about how these sex steroids may impact mitochondrial function, and we tested different concentrations of cholesterol, estrogen, and testosterone to see if they would have any effect on protein's function. And we actually observed significant differences in our experiments. The presence of estrogen actually increased OPA1 mediated membrane remodeling, whereas presence of testosterone significantly reduced OPA1 ability to oligomerize and remodel these membranes. We also see a similar pattern in membranes where, again, the presence of estrogen increases CRISPR number versus we don't see the same effect when we use testosterone. So with that, I want to thank people in my lab. Siri and Akil did the majority of this work. And I also want to thank the other members of our lab and our collaborators, specifically Matteo and Luciano, who performed MD simulations. Adam Frost's lab provided us the cardiolipin probe, Dale's lab, and our other collaborators. And thank you.
Wonderful. Another excellent talk. Thank you, Halil. I can't really state how exciting it is to see that little, collection of black burr underneath that membrane interacting loop, just kind of proving that the membrane interaction loop is where the clustering of the cardiolipin is happening. It's a bit counterintuitive to think that that's an amazing observation in an experiment, but it truly is, because you would imagine that, packing defects or other things might be felt through the membrane. It doesn't mean that those lipids cluster underneath the loop. So again, some really, really beautiful detailed work. And now I just invite, if there's any questions from attendees, please raise your hand. And I would also reiterate that given the timeline as well, that Dr. Aiden and Dr. Masud put their email addresses in the webinar chat. If you'd like to interact with them directly afterwards, please reach out. So are there any questions? Oh, we've got our first hand here. So Binks Wattenberg, I'll go ahead and allow you to talk. Please unmute.
Okay, thanks very much. These are both great talks, and there's an intersection, and maybe I missed something here, but from the last talk, it seems like the cardiolipins with saturated chains are not as efficient as binding and utilizing OPA1. But in Dr. Masud's talk, the mutation of her ABHD protein would actually prevent the introduction of saturated chains into cardiolipins. So, and yet it seems like that, at least at the organismal level, everything is working okay. So I would invite comments from either or both speakers about how both can be true.
I can mention this quickly. So cardiolipin actually interacts with many proteins in mitochondria, and to me, it's actually the real master regulator of mitochondrial function. And that's one. The second one is, if you look at the changes in crystal morphology, OPA1 is not the main player. Actually, it's the Micos complex. If you mutate Micos proteins, you see a significant change in crystal morphology. But if you look at OPA1, it's not as you don't see a stark difference. The crystal morphology only changes 10 or 15%, and we show this, and other groups show that as well. So one explanation might be is that OPA1's primary function is membrane fusion, not the crystal remodeling, and those processes are sensitive to changes in crystal morphology.
Yeah, as a quick follow-up, I wonder too if the position, so the MLCL, the other reality is whether or not it's taking place at what position on the 4 acyl chains, because it seems like the flux through phosphatidic acid to phosphatidylglycerol, some of the cardiolipin backbone is set by the precursor lipids that are being used to synthesize it. And the site of remodeling, it is chain specific. So perhaps too, it's just how granular you get with the species as well. But I wondered if the incorporation of unsaturation, is that just another sign that it is about fluidity, packing defects? Because the only other thing I could maybe think of in the stiffness is that the MLCL might have a bit less of that, interleaflet interdigitation or communication, but it's hard to really nail down these things in an experiment.
Well, we really analyzed that carefully, actually, what the chemistry would be. And one of the explanation is that, missing that acyl chain really changes the head group orientation. And that might be the reason why the remodeling is restricted. And OPA1 is very sensitive to that. We actually have a point mutation in our lab within that loop where the protein binds and oligomerizes perfect normal on nanotubes. These are already like curved membranes, but it cannot remodel liposomes. So it cannot overcome the energy barrier required for remodeling the membranes, but that doesn't mean that the oligomerization is impacted. So there are really few key nuances at the molecular level.
But that's so fascinating. And again, just to close, I really can't state enough how great it is in your papers where you actually see the remodeling assay. Does it tubulate and bend the lysosomes? Because you can see how you need to look at, all of those angles. It's not just whether it's recruited or binding. So with that, once again, thank you very much, Dr. Aden, for a great talk. And once again, thanks to Dr. Masud as well. So I've been your host, Dr. Joshua Pemberton, at the University of Western Ontario. Thank you very much for joining us for the Lipid Research Division Seminar Series. And I'd like to also thank Kalsum with ASBMB for the technical help and 150 odd individuals for taking time out of their summer day. So thank you very much. And hopefully you'll join us again in September when these start up again.
Thank you, Josh. Thank you. It was great.
Thank you,
We'll see you later.
everyone.